For this opportunity. So I'm going to talk about the development of epigenetic biomarkers for European sea bass aquaculture. We're going to concentrate on markers for temperature treatments. But first, let me explain a little bit what is epigenetics. No, so I don't know the background of the audience, but I'll try to be as clear or as didactical as possible. No. So the epigenetics is defined as the study of phenomena and mechanisms that cause this. I'm going to explain in a minute what this means, chromosome-bound, mitotically or meiotically heritable changes that do not, changes in gene expression that or phenotype that do not depend on the changes on the DNA sequence. So epigenetics has had a lot of attention lately, and it tends to try to be given the power to explain everything, but this is not the case for sure, no? So if we have the DNA molecule, one DNA molecule is the genome that we all have, and this consists of its stored information. And then we have this DNA molecule that is packaged into associated proteins all together form the chromatin. And this information, this genomic information is organizing somehow. And whether these genes are expressed or not in different tissues or how they are regulated is the source or is what explains that the same genome that we have in our cells is able to give different epigenomes. So that means different cell identities or different cell types. So we have in all our cells the same genome, but we have different epigenomes, in our case, more than 200 different cell types. So this is thank you to the epigenetics, no? Which is not changes in the DNA sequence, but changes in the how this sequence is read and therefore how these genes are expressed or not. So there are, let me explain that there are three major epigenetic mechanisms. No, one is DNA methylation, the other one is histone modification, and the other one is non-coding RNAs. The three of them are important and they act together. But because DNA methylation is the most genetic of the epigenetic mechanisms, so to speak, because it is an actual change, not in the nucleotide sequence, but also. But in the DNA itself, methylation of the cytosine, carbon 5 of cytosine, this is the most studied one and the most easy to perhaps to measure. No, one of the most easiest to measure, and therefore for sure is the one that in many areas, not aquaculture but evolutionary biology, for example, in ecology, is being used. So I'm going to go into detail into this one, but remember that there are three, and you can have biomarkers for the three of them. In fact, in non-coding RNAs, also there is a lot of effort being done now in aquaculture-related issues. So the important thing here, as you can see, I don't know if you see my cursor, but here you have epigenetic variation that depends on two things: one, genetic variation, and the other one, environmental variation. And this environmental variation can induce epigenetic changes that are controlled also by genetic variation, which condition them, and altogether genetic and epigenetic variation control gene expression. And this gives rise to the phenotypic variation or the traits of interest that we observe in our fish. So let's focus on DNA methylation as an epigenetic mark. So you have here a sequence, ACGTG, for example, and the last G is changed. This last nucleotide is changed to a T. This is a mistake here. This is a genetic change. This is a mutation, what we know as a mutation. But epigenetics are not mutations, are simply modifications or called epimutations, but different from genetic mutations. Here, the sequence, as you can see here, does not change. The sequence is the same here or here. What only changes is the methylation of this cytosine. And this is represented, the line represents the DNA sequence. For simplicity, is simply referred as a line. And the black dots and the white dots represent places where this DNA sequence can be methylated or not. And if it's methylated, it's represented usually as a filled black dot, and if it's not methylated, as a white dot. So for example, here below, we have a sequence of DNA, and this one, you have 4 places where it could be methylated, but it's methylated only in 3 of them. So you have 3 black dots and 1 white dot, no? So for representation. So we would say that this particular fragment of DNA sequence is methylated 75%. Methylation goes from 0 to 75%. And the good thing is that when promoters are methylated, usually this shuts down the expression of the gene. So no methylation in the gene promoter means the gene off. Methylation of the gene promoter means the gene off, a gene on, off. And here you have the relationship between more DNA methylation and less gene expression. It's usually an inverse relationship, although exceptions exist. So then, when we know the level of methylation in one or different loci in the genome, here, for example, we have one gene and we have 3 regions of the gene, each one in different, what's called CPGs. This means CPG is the piece for the phosphate bond, so it's the cytosine and guanine. So the genome can only be methylated in the cytosines that are next to a guanine. So these are the level of methylation from 0 to 100%, and these are CBAs exposed to different temperatures. So these ones, they have ones exposed to lower temperature. In this particular case, in this gene, they have higher levels of methylation. Well, these are epigenetic marks. The level of methylation and the difference in methylation between one type of fish, the one exposed to low temperature and the ones exposed to high temperature, it can be used as an epigenetic mark to identify whether these fish have been exposed or not. And this has, of course, epigenetic, expression consequences in terms of how these genes function. So, important thing about the epigenetic marks is it's like writing in a pencil, no? You can write this information in the genome, you can read it, you can copy it, and you can erase it. So for example, there are enzymes called DNA methyltransferase 3, which is the de novo methylation that convert an unmethylated region of the genome into methylated one. And then we have copiers, which are another type of enzyme, DNA methyltransferase 1, that this is called the maintenance DNMT, which what does is takes one fragment of DNA which is methylated in whatever the sequence, and then when there is mitosis, and so the cell divides into 2 daughter cells, these marks are carried into the daughter cells. And then we have readers that can interpret this methylation, and these are methyl binding domain proteins, no? Methyl complex protein 2 or methyl binding domain protein 1 or 2, for example. And then this DNA that is methylated can be also erased. And this can be done in 2 different ways, either active methylation by the action of these enzymes called 1011 translocation deoxygenases, or simply by passive demethylation, by dilution and not concur of the DNA methyltransferases. So epigenetic marks is like a code that can be written, read, copied, and erased. And during development of the organisms, these epigenetic marks change. And there are some changes that are common, some changes that are not common. Let me explain this. So we have here the level of methylation from 0 to 100%. And here we have 3 different species in fish. This has been studied only in very few species, Medaka and zebrafish, basically 2 models. We're doing now these studies in the sea bass. And you have the levels of methylation this is in the sperm and this is in the eggs. In zebrafish, the sperm goes demethylated and the egg goes methylated. They reach a middle point here, and this is the profile of methylation. In the mouse and in the Medaka, the other fish, there is this first wave of demethylation. This is less pronounced in zebrafish. And then there is a remethylation, and this affects the whole animal, everything. But then at one point, there is methylation of the somatic cells, and it stays more or less constant the rest of the life. But then during time of the gonad formation, there is a demethylation of the PGCs, the primordial germ cells, which will give rise to the spermatogonia or oogonia. And then finally, during gametogenesis, in the case of males, during spermatogonia, a renewal, and then there is changes, very small changes I can put here in methylation. So there are 3 places during the development of an animal where you can reprogram these cells, these epigenetic marks. So it means that during early development, for example, this is the marks of the sperm that are all erased and then rewritten again, and then erase again. And this is during embryogenesis. The second time takes place during gonad formation and sex determination. And the third place is during gametogenesis. These are the 3 windows where we can manipulate the environmental conditions in order to change epigenetic marks. So for example, in the sea bass, some years ago, we did a manipulation of temperature during early development. So this is around 30 days, 60 days. And when you change the temperature here, what you are affecting is the changing the epigenetic marks of the somatic cells. In that case, of the somatic cells of granulosa and thicker cells of the gonads, no? The females and the males, Leydig cells and Sertoli cells. And this affects then how this animal will later, during development, undergo the process of sex differentiation. Or for example, if you want to program the fish to be more responsible to the substitution diets, then this is the sort of reprogramming that you have to do at the somatic cells during the renewal of the cells that will give rise, for example, to the epithelial of the digestive tract. But if you want to pass these genetic modifications to the next generation, then you have to do the manipulation very early during development, or at least at the time of the germ cell reprogramming, okay? So then we can talk about intra and transgenerational epigenetic changes. So mitotic inheritance means that when one cell divides, then and forms two daughter cells, whatever are the marks that this cell has will be passed to the following generation in terms of the cell generation, so during mitosis, okay? But during meiosis, if the animals escape or there is changes that you induce at this time, imagine, for example, that you expose the animal here to a condition that normally doesn't experience, for example, smoking in a pregnant woman, this can have an effect on the offspring. So this is what's called the transgenerational epigenetic effects. But in the case of a human or a mammal, when the exposed organism, the mother, the P generation is always the exposed generation, is exposed to a chemical, for example, a contaminant, it is exposing actually the fetus that carries in the bum and the germ cells of the fetus. The fetus will be the son or the daughter, the F1, and the germ cells will be the grandchildren of this woman. So this will be directly affected by in this case, by the chemical of the smoke. So in order to have epigenetic transgenerational inheritance, you need to prove that this is epigenetic change in the germ cells, and this will be only accepted as transgenerational effects if they are visible in the F3, because this is the first non-exposed generation. In the case of fish, which are egg-laying, not viviparous, or the majority, then when you expose to a fish to a chemical or to temperature or something, you will affect the fish and the germ cells of the fish. And then these germ cells will become the F1. And then if the F2 is not affected, then you can speak about transgenerational effects. This is the first non-exposed generation. So always for epigenetic transgenerational effects, you need to look at these effects whether they are or not present in the first non-exposed generation. In the case of fish, the F2. In the case of mammals, F3. But then we have that the aquatic animals are very well suited for epigenetic manipulation. Why? Because if we look at fertilization and early development, we see that in mammals, both things are internal. They take place inside of the mother. In the case of the birds, fertilization is internal, but then early development is external. The egg that is incubated by the chicken, but still you can manipulate this temperature. But in the case of mollusks, crustaceans, or fish, everything is external. So therefore, these animals, the aquatic animals, are very well suited for epigenetic manipulation, better than other type of livestock. And this means that, as I explained before, there are windows of development during early development, which is gamete development, fertilization, embryonic development, and post-hatch birth development, whether during these windows of sensitivity, these animals are very sensitive to any sort of environmental influences, biotic and abiotic. And this can have long-lasting consequences in growth, product quality, health, welfare, and reproduction. And what are these sort of changes? No? So we know that epigenetic marks can be, or epigenetic the profiles can be modified by changes in the diet, by the level of stress, by temperature, by the amount of exercise, by the presence, of course, of pollutants. These are all external factors, here put in green. Or also it depends, epigenetic changes also are depending on the genetics. I'll show you before that there is a genetic component on the epigenetic profile, so we don't have to forget that. We have to take into account that epigenetic marks are tissue specific, that also can be sex specific, and on top they can change with age. No? So when we compare intrinsic and extrinsic changes that can be induced in an animal, and we compare these changes, whether they're genetic or epigenetic, we see that both of them are preserved in mitosis. So during cell division, of course, the genetic information is maintained, but also is maintained the epigenetic information, can be preserved in meiosis, also not only the genetic, but also the epigenetic if it escapes reprogramming. In contrast to genetic, the epigenetic is cell specific or cell tissue specific, and in contrast to genetics is responsible to environment. So we have to take advantage of these characteristics. Now let me give, to finish, a couple of examples of what we have been doing before or in the context of this project, no, in the PerformFISH. In this case, we look at different levels of methylation, combining males and females, fish exposed to low and high temperature, and we build sort of a key that is able, when applied this information, when we know these different loci, each one of these marks is the level of methylation, in this case the difference, in a specific loci in the genome, we're able to construct a model that is able to predict the sex of the future sea bass with a success of about 90%. And this has been has been patented as something that can be useful. No? But then these epigenetic marks, if they survive mitosis for a long time, then can be used to infer, and this is where I'm going with what we did in the PerformFISH project, they can be used to infer the environment and the quality of the environment that the animals experienced in the past. So this is an exercise, no? We look at metastable epialleles, which are epialleles are changes in the methylation in different alleles that are metastable. Metastable means that the same change is present no matter what tissue you look. I told you before that the epigenetic markers are tissue specific, but if you infer the changes or induce the changes when the tissues have not been differentiated, so during very early development, embryonic development, then the changes are in all tissues the same for equal. So these are what's called metastable epialleles. And if you combine them with pure epialleles, which means that no matter what the genetic basis, I told you also before that the epigenetic changes is dependent in certain degree into the epigenetic background. Well, there are some of them that no matter what is the epigenetic background, you always get the same response. So these are called pure epialleles. So if we combine these, we can infer the past, no? And this has been published, no? And now what we're looking, not only us, but other people too, is looking at how these epigenetic changes can be used to address aspects of phenotypic variation. And in aquaculture, this is related to what's called the key performance indicator, that usually are things like survival, growth, reproductive fitness, and so forth. No? But also, this is important in ecology and evolution, and then you can change key performance indicators for key ecological indicators or key fitness indicators. No? So in short, this is how species are prepared to deal with new environments. No? And if we look at epigenetic changes that are linked to phenotype. So one phenotypic change, we can say this will induce this certain phenotype, and this is pure, meaning that doesn't matter what genetic background we have there, we always see the same, and on top is heritable, so can pass from parents to offspring. Then we can use these changes to select fish beyond the typical genetic selection. So for example, we can ask our question like, let's say this is like data that we have also from a different project, but to illustrate the potentialities. we can say, okay, we want of within the fish, the broodstock fish that has been previously selected, let's say, for example, for growth, let's try to see which ones, what families give the best growth, not only this, because within the selected fish, still going to be phenotypic variation, but on top, the ones that they have higher survival, and on top, the ones that they have less deformities. So we can build an equation in which we give different weights to these characteristics that we want, and then we can look at the epigenetic marks. Remember that these fish genetically are very similar. We can look at the epigenetic marks that explains phenotypic variation. I can build a model using intelligence on artificial intelligence or machine learning, and then we can separate, as you can see, the red balls and the green balls represent fish, each one a sample, that are what we consider the good and the bad fish, and this prediction accuracy is pretty high. No? So, and this epigenetic selection can be combined with classical genetic selection, in where then you select the broodstock based not only on genetic information, but on epigenetic information. And then the epigenetic information or the epigenetic manipulations you can do, you can manipulate the broodstock to affect the larvae and juvenile, or you can manipulate the larvae and juvenile or embryos to influence the adults. No? So here in the PerformFISH, what we did is to expose fish during very early development from epivolley to first feeding to 3 different temperatures, all right? And then we submitted, left the fish at the same temperature for the rest of their lives. And then we look at different genes that were differentially expressed, so the DAGs here at 14 versus 17, or genes that were differentially expressed between 20 and 17. 17 was the middle temperature used as a reference. And also we look at genes that had differential methylation loci or cytosines, okay? And we can see that among these, there are genes that both they have epigenetic and transcriptomic differences, okay? And even we can go further and select the ones that they respond no matter whether the temperature goes down or goes up. And we can build them like graphics like this one, in which this is the methylation level at the 3 different temperatures. And here we have a group of genes that each one of them, they follow exactly the same pattern. We can do, there are genes, of course, that they follow exactly the opposite pattern. No, but in this case, I just to illustrate, I put here genes that the methylation of which goes up with temperature, okay? And then we can look at the expression, we can look at the Go terms, and we can build a panel of genes that they have epigenetic changes, represented by E, transcriptomic changes, or both of them, no? B. And we can look at the 14 versus 17 degrees, 20 versus 17 degrees, and then we get this, for example, these are examples, no, of genes that they have epigenetic changes at these combinations of temperature, or transcriptomic changes, or both, no? And that can be seen at very short-term effects, so first feeding, during juveniles, mid-term effects, adults, long-term effects, and 3 years old, very long-term effects. So that means that if we are able to find what epigenetic indicators, markers, we select by looking at fish like very early or in juveniles, we will be able to infer, to guess, to predict which of these animals or which of these stocks will have effects on gene expression when adults. So, and this is what is called the epigenetic marks with both diagnostic and prognostic. So imagine that we have developmental time here in this arrow, time 1 and time 2. And here we have the control and the treated. Treated can be treated with temperature, with an artificial diet, with a parasite, with a pathogen, sorry, whatever you want, okay? So imagine that the gene expression in the control, of course, does not change, but it changes. This treatment, whatever it is, affects the expression of this gene, no? But this change is only seen at time 2. The beauty about epigenetic changes is that some of these changes are already present at time 1, when you still do not see changes in the gene expression. So you have then found epigenetic markers that they have not only diagnostic, but also prognostic value. Diagnostic because the changes are visible in T2, and prognostic because the changes are visible in T1. Okay, when in terms of phenotypic traits, you can still not see anything. Okay, so you can predict what's going to happen. This is keratin associated protein 10.4 is an example that we provide here. So at 5 days of age, so very early during larval development, fish exposed to low or 17 or 20 degrees, you don't see changes in the expression of the gene, but you already see changes in the methylation of this gene, changes that persist in a significant manner at 479 days or much later. And then, of course, these changes, in this case, an inverse relationship, are seen in the gene expression. So this is a way, so measuring the methylation of this gene at day 5, I can tell whether what's going to happen at day 479. Okay, so it's a way of predicting. And so the conclusions for this is that the epigenetic mechanisms integrate both genomic and environmental information to shape the phenotype, so the characteristics that we want to see. Oh, sorry, this is and jump one. Epigenetics have a promising future in aquaculture because aquatic animals are very sensitive to epigenetic manipulations when compared to, for example, birds or mammals. We report, we think that is the best of our knowledge, the first identification of epigenetic biomarkers with both diagnostic and prognostic value that can anticipate the long-lasting effects of thermal alterations, which we know that these alterations during early development have phenotypic consequences in terms of like the fitness or the growth of the animals. No? And we pave the way for similar developments for other pharma species. And then results highlight the importance of an accurate maintenance of environmental parameters, particularly temperature during SIVA's early development, and the power of DNA methylation marks as diagnostic and prognostic tools. And here I jump this one that says that what I explained before, no, that we have developmental windows in which the animals are very sensitive to environmental perturbations. And one thing that I forgot to mention is that in contrast to other sorts of manipulations, for example, hormones and so forth, all the epigenetic changes are essentially dealing with manipulation of environmental factors or the diet. And this is in line with the concept of sustainable aquaculture. Just let me tell you that we are preparing a book on epigenetics. I explained today only very few parts, but there is a lot of studies are going on now on skeletal muscle growth and nutritional programming, very important for aquaculture, epigenetics and disease, stress response and immune function, sex determination and differentiation, and then also not only in fish but also in other farm animals, no? And epigenetic biomarkers and how they can be integrated with genetics in breeding programs. And to finish, I just want to thank the people of my group, and particularly Alejandro Valdivieso and Nuria Sanchez Baizano, who did the study that now is submitted to aquaculture, our collaborators in France and in Greece and other places, the funding, and particularly very much the funding of the PerformFISH. Thank you for your attention.